Hi, my name is Eric Soy. I'm based out of Cold Spring Harbor Laboratory, and I'm the executive editor of Life Science Alliance. I'm joined today by Charles Bourque from McGill University. Welcome, Charles.
Hi, Eric. Nice to be here.
Thanks for coming. So your lab works to understand how the brain controls osmotic homeostasis. And we know that maintaining this homeostasis is a vital process because even acute changes in osmolality can cause a gradient of neurological symptoms, which can ultimately lead to death. First question is really, what sparked your interest in this area of research?
Well, a very long time ago, when actually in graduate school, I was confronted with the decision to decide what my thesis project would be, and I was prompted by my supervisor to ask the question: how does the brain detect changes in osmolality? And as a graduate student in physiology, it was shocking to me that a question like that would be even asked, because I had learned in textbooks that, there are things called osmoreceptors, and that, there's a monitoring ongoing of blood osmolality, and then you activate all these homeostatic responses. So it seemed clear to me that this was all understood.
Mm.
However, looking into it, it turned out that the textbooks reported these homeostatic things, but it was really, there was a big black box between how does a change in osmolality lead to homeostatic response. And it opened up to me the whole idea that these things could be studied at the cellular and molecular level to really understand the physical basis for this kind of process. And so, of course, this is a huge area, and so it kept me busy for a couple decades.
Great. So getting into your work on sleep and the signaling that controls body temperature, can you explain the anticipatory period and why it is important?
So, the part of the day where we're the most challenged from a hydromineral balance or osmotic point of view is while we sleep, because although our brain detects that we're gradually dehydrating as we evaporate water just from breathing, um, our brain doesn't wake us up to go and drink water and keep that adjustment going. So other things are needed. And one of the things that happens is that the brain gradually increases the amount of vasopressin secretion. Vasopressin is an antidiuretic hormone, and it instructs the kidney to gradually reabsorb more and more water as you sleep. So that's a very important homeostatic response that we're very interested in studying also. But the anticipatory period is something that had been observed for several decades in different species of rodents, and it was an intriguing kind of thing that there's this period where there's an increase in water intake just before the animals go to sleep, and there's a similar peak at the beginning of the active phase, and of course at the beginning of the active period of the day we think, well, okay, we're waking up, we are slightly dehydrated, maybe we eat, so we want to drink. But what makes us drink at the end of the day? And so, although it had been proposed that this could be a mechanism to try to create a water reserve overnight, it had never really been carefully examined using experimental approaches to see what was driving that period of increased water intake. And so with experiments that were done by Claire Gazowski, who was a PhD student in my lab at the time, she looked to see if that increased water intake was motivated by something physiological. So we know that water intake is stimulated by increased body temperature, for example. So she looked to see if there was an increase in body temperature at that time, and there wasn't. And she looked to see if blood osmolality was high for some reason, and it wasn't. And so she looked at every parameter that we know can underlie a physiological drive for thirst, and could find nothing. And so this left the notion that, well, this is exactly the kind of thing that the central clock might be doing, anticipating that, okay, you're about to go to sleep, it'd be a good idea to create a water reserve. And so she tested that hypothesis in her thesis, and showed that the clock indeed is responsible for driving that water intake. And so, and more than this, she showed that if you deny the animal that increased water intake, there's a cost to it, that you are more dehydrated the next morning. So it serves a physiological purpose. So we think it's one of the better examples of a circadian rhythm that serves an important functional physiological role.
And do we know, is this anticipatory period, is this all light dependent, do you think, or is it based on a predictable rhythm based on, schedule set by previous days?
So it's a rhythm that persists in constant light conditions or constant darkness, and is a classic. It occurs always at the same relative time to the relative to the light cycle. So it is driven as a clock mechanism.
Mm-hmm.
And our experiments showed the circuitry between the suprachiasmatic nucleus and the thirst driving neurons in this small structure in the preoptic area called the OVLT as being sufficient to drive that rhythm. So it's driven by the clock as a clock dependent process.
I see. So is it known, or do you have any insight into if mice were kind of set off that schedule, moved off that schedule, if they were sleep deprived, for instance, do we know if the anticipatory period would extend, kind of hopeful for that eventual sleep, or is it a fixed time regardless of when actual sleep occurs?
So we haven't looked at that, but our data indicates that it's really that these SCN vasopressin producing neurons seem to turn on like about 2 hours before the lights go on, which is when the animals transition from the active to the sleep period. And the thirst drive is driven by that small increase in activity at that time. And so you would hypothesize then that anything that perturbs when that increased activity occurs in the clock would also lead to that so-called anticipatory event. So this is a really cool idea, but it hasn't been tested experimentally yet.
Okay. Now, yeah, comparing these, and I want to get the terminology right, so correct me if I'm saying this wrong, but the scheduled versus unscheduled osmoregulatory effects. So you just described scheduled effects, where the increased water intake before sleep, you've shown is caused by the SCN vasopressin neurons. They project to the thirst neurons in the OVLT. But can you talk a little more, what you just alluded to, about the unscheduled effects or responses, and what happens there, and how you model that?
Right. So the neurons that are being turned on by the clock a couple hours before sleep, which leads to this anticipatory water intake, those same neurons are the ones that are we think monitoring blood osmolality. And so they have an ear to the clock, and if the clock tells them go drive, they'll drive the water intake. But those neurons are also sensitive to changes in blood osmolality themselves. And so they are like dynamic sensors of blood osmolality. And so if you have a salty meal, a salty meal is enough to bump your blood osmolality by a few points, which on its own is not life-threatening or anything, but it is a homeostatic challenge, and so it does trigger thirst, and it does it by activating these neurons. So this is what we call an unscheduled effect, because this can happen at any moment of the day. If the animal experiences a salt load for some reason, either an experimenter injects sodium into your system, or if you, like I said, you either you eat a salty meal, which increases your osmolality, or if you drink a bunch of water and dilute your blood such that your osmolality decreases, this will suppress any basal thirst you have and activate other homeostatic responses. And so these things happen throughout the day, and so these are the things we called unscheduled. And what was remarkable by this finding is that the clock is able, because the clock is connected to all these homeostatic pathways to be able to drive these homeostatic responses. We thought, well, maybe these sensor neurons that take care of unscheduled responses are also capable of driving these homeostatic responses through the circuitry of the clock. And so this is the kind of an important part of the discovery, is that the clock can mediate these unscheduled responses because it's wired to do it normally via the timer. So it's like a timer that regulates your, like we have a timer in our living room that turns a light on at 8 p.m. Well, there's a little switch on that timer that allows you to manually turn it on anytime you like, right? So it's the same idea.
Yeah, that's really interesting. And then as far as in people and what sort of things in life that cause these unscheduled effects. So, you know, you mentioned just even drinking high levels or intaking high levels of salt, or conversely high levels of water to skew that balance. Are there some disease related aspect of this where this occurs in people, that this might be the underlying signaling?
So there are people who have defects in the ability to detect osmotic perturbations.
Mm-hmm.
And what happens in those cases is that, for example, if you if your body allows itself to become hyperosmotic because you're not detecting it, this will naturally draw an increase in water absorption in all the cells, and ultimately will increase your blood volume. And then we have other sensors in the body that'll pick up this increased blood volume and activate similar homeostatic responses to say, whoa, blood volume's too high, so we have to, you know, tell the kidney to excrete. So there's a strong parallel between different homeostatic systems that can achieve kind of the same thing, but in a different way. And so you might find yourself now at a different set point with regards to osmolality, but you're still alive. So you know, there's enough redundancy in the system to take care of these things. I guess there's two interesting aspects here. One is that the clock controls these things to keep, you know, to do these scheduled adjustments, and people who lack the scheduled adjustments, and there are people who have these disorders, they experience things like nocturnal polyuria. So, you know, these people don't have the mechanisms that tell the kidney to reabsorb the water, and so instead of making it through the night without dehydrating, these people become dehydrated overnight, and their bladder fills up, and it wakes them up, and it perturbs their sleep. So a lot of people who have perturbed sleep, you know, after long investigation with endocrinologists, is discovered that, oh, you lack the circadian rhythm that increases your blood vasopressin. And so they are then prescribed with a pill that, you know, can activate vasopressin receptors, and they take those before they go to bed, and it fixes their problem. But it's a great example of how circadian rhythms not only are important, but if you don't have them, there's a pathology associated with it.
Yeah. Wow. Well, this is fascinating work, Charles. So I thank you for sharing it with us and giving us more details on the ongoing research. I think we should wrap it up there, and I appreciate you taking the time to talk with us.
Well, thanks very much. It was a pleasure talking to you.
Okay, thanks.
Take care.
Out to the